أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين.
Assalamu alaikum. Greetings of peace, mercy, and blessings. This video lesson is for grade 10 students, but of course, everyone who is willing and interested to learn is welcome to join us. Today, we will talk about one of the most interesting questions in science. How is DNA made out of a DNA template? How is RNA made out of a DNA template? How is protein made out of a RNA template? And how does mutation result to change in the structure and functions of protein? Are you ready to answer these questions? If yes, make sure that you have your paper and pen ready so that you can write all the things that you have learned from this episode. I am teacher Sandra Calderon Amino, your science teacher for today. You have learned in grade 9 that many genes in plants and animals behave differently than the genes that Mendel studied in peas, where traits are not entirely controlled by dominant and recessive genes. You also learn how the genes in your deoxyribonucleic acid DNA influence your characteristics. DNA determines things such as your eye color, your hair color, height, and even the size of your nose. Since the rediscovery of Mendel's work in 1900, the definition of the genes has progressed from an abstract unit of heredity to a tangible molecular entity capable of replication, transcription, translation, and mutation. The central dogma of molecular biology is an explanation of the flow of genetic information within the biological system. It is often stated as DNA makes RNA and RNA makes protein. Before we start, work on the initial assessment activity to measure your understanding of DNA and RNA structures. Activity number 1. Getting to know the DNA and the RNA structure. Objective: compare the structures of DNA and RNA molecule. Material: activity sheets. Procedure: number 1. Listen carefully to the given information about DNA and RNA. Component molecules. 1. The DNA molecule is composed of 3 types of component molecule: phosphate group, the sugar deoxyribose, and the bases adenine, thymine, cytosine, and guanine. Number 2. There are 3 molecules that form the basic building block of DNA, the nucleotides. Each nucleotide is composed of 1 phosphate group, 1 sugar molecule, and 1 of the 4 bases. Across the strand of the helix, adenine always pairs with thymine, and guanine with cytosine. RNA, ribonucleic acid, like DNA, is a nucleic acid. However, RNA structure differ from DNA structure in 3 ways. First, RNA is single-stranded. whereas DNA is double-stranded. Second, the sugar in RNA is ribose. DNA has deoxyribose. Third, both DNA and RNA contain 4 nitrogenous bases, but instead of thymine, RNA contains a similar base called uracil. The uracil pairs with adenine. The major types of RNA include messenger RNA, ribosomal RNA, and the transfer RNA. Number 2. Fill in the comparison table below. Guide question number 1. What are the components of DNA and RNA molecule? Number 2. What is the structural difference between DNA and RNA? Number 3. What nitrogenous base is found in RNA but not in DNA? How is the pre-assessment activity? Did you find it hard? You have nothing to worry about because we're going to elaborate on this throughout this episode. Are you ready? Well then, prepare yourself and enjoy watching while learning. Let's take a closer look at DNA replication. DNA is made up of many nucleotides, each containing a base represented by the letters A, adenine, T, thymine, C, cytosine, and G, guanine. A piece of DNA contains 2 strands of nucleotides twisted together to form a double helix. Each strand has a 5' end and a 3' end. The 2 strands run in an opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and result in the formation of a replication fork. The separated strand each provide a template for creating a new strand of DNA. An enzyme called primase start the process. This enzyme makes the small piece of RNA called a primer. This marks the starting point for the construction of a new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction from 5' end to the 3' end. One of the new strand of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to the 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore make a strand in a series of small chunks called Okazaki fragments. Each fragment is started with a RNA primer. DNA polymerase then adds short row of DNA bases in 5' to 3' direction. The next primer is then added further down in the lagging strand. Another Okazaki fragment is then made and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers for both strands of DNA. Another DNA polymerase enzyme then fills in the gap that are left behind with DNA. Finally, the enzyme DNA ligase seals the fragments of DNA in both strands to form a continuous double strands. The following events can help you understand the process of transcription. When a gene is switched on, an enzyme called RNA polymerase binds and opens the DNA molecule that will be transcribed. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which free bases are added to the messenger RNA. As the DNA molecule opens, the RNA polymerase slides along the DNA strands and links free RNA nucleotides that pair with the nitrogenous bases of the complementary DNA strand. When the process of base pairing is completed, the RNA molecule breaks away as the DNA strands rejoin. The RNA leaves the nucleus and goes to the cytoplasm. This process is called transcription. The following events in translation can help you understand the process. Step 1. As translation begins, mRNA binds to a ribosome. Then, tRNA molecules, each carrying a specific amino acid, approach the ribosome. The tRNA anticodon pairs with the first mRNA start codon, arginine, uracil, guanine, to form the initiation complex. The two molecules temporarily join together. Step 2. Usually, the first codon on mRNA is AUG, which codes for the amino acid methionine. AUG signals the start of protein synthesis. Then, the ribosome slides along the mRNA to the next codon. Step 3. A new tRNA molecule carrying an amino acid pairs with the second mRNA codon. Step 4. When the first and the second amino acids are in place, an enzyme joins them by forming a peptide bond between them. And step 5. As the process continues, a chain of amino acids is formed until the ribosome reaches the stop codon. The polypeptide chain is released. Protein synthesis is complete. Activity number 5. Trace the code. Objective: identify the amino acids coded for by the mRNA codon using the genetic code table. Materials: genetic code table, activity sheets. Procedure: copy and fill in the table. Refer to the genetic code table to identify the amino acid. To determine the order of bases in the first column, which is the DNA, and the second column, which is the codon, and the third column, the anticodon, consider the complementary base pairs in DNA. Adenine pairs with thymine, and guanine pairs with cytosine. While in RNA, adenine pairs with uracil, and guanine pairs with cytosine. To identify the amino acid, look at the bases in the mRNA codon. For example, AUG, using the genetic code table, look for the first letter in the mRNA codon on the left side of the genetic code table, A, and the second letter in the mRNA. On the second letter column, look for letter U, and the third letter on the right side column for letter G. Here, AUG codes for the amino acid methionine. Do the same with the other codons in the chart. Question number 1. Why is specific base pairing essential to the processes of transcription and translation? Question number 2. How many codons code for one amino acid? Now, let's test your mastery of the lesson. Answer the summative assessment. Direction. Sequence the following steps in protein synthesis from first to last. A. Transcription. B. tRNA, amino acid units links to mRNA. C. Amino acid separate from tRNA. D. Polypeptide chain assembled. E. mRNA links to ribosome. F. Stop codon encountered in mRNA. Let's check your answer. For the summary of the lesson, let's sing the song about the central dogma.
DNA to RNA, RNA to protein, the central dogma song. From DNA to RNA, RNA to protein, it's almost never wrong. Relate in control of your traits, replication. It makes the DNA copy semi-conservative, and it's not that sloppy, but just in case we got some exonuclease. Haters get mad cause I got DNA polymerase. In transcription, we unwind with helicase, RNA polymerase, TTU we replace single-stranded RNA, add a cap and polyate to the cytoplasm, from the nucleus away, subunits of ribosome, surround it and read it, tRNA they need it, initiate.
I hope that you have learned new lessons today. Thank you so much for speaking with me, and see you on the next episode. Masalam!
Baby, your cellular construction has DNA instructions for base simple co-directed peptide production. Quick Spanish deduction started from induction, a simple path to follow. DNA to RNA, RNA to protein, the central dogma.